What we found by analyzing some recent literature is, in the most case, soybean land races and accessions from East Asia have the functional E1 gene, while as you travel north you start to see some occurrence of the missense E1AS and the two E1 null alleles. So accessions from East Asia often have functional E1. In North America there's quite the contrast, however. Instead, although you can make a line that is adapted to, let's say, northern Missouri with functional E1, breeder selections have shown that the vast majority of lines north of about 36 degrees latitude contain the missense E1AS alleles, while south of that 36-degree latitude line, nearly all soybean lines contain the functional E1 alleles. As I said, you can make soybeans that will mature north of that line with the functional E1 gene, but as I've talked with Dr. Kober about, it seems like those lines are tall and lanky but low yielding. We also have a system of maturity groups that helps growers identify the best lines that are adapted to very narrow bands of latitude in their areas. So for instance, in my location in Missouri, in the center of the graph, our growers grow maturity group 3 and maturity group 4 lines, while in northern Minnesota they might grow double zeros, and in southern Arkansas they might grow maturity group 6s. This is the U.S. maturity group system. It's worked fairly well over the last 50 years or so, and it's just being fine-tuned now as climate change is affecting some things and growers shift to different systems, such as they do the early production system in the South. I also want to talk about the plant growth architecture genes. We have indeterminate, determinate, and semi-determinate soybeans. What this means in this figure is that indeterminate soybean grows tall, and that's because after flowering, vegetative growth continues along the main stem. This is shown by capital DT1, indeterminate, plants continue growth of the main stem after flowering. Determinate lines, shown in the figure here, terminate main stem growth shortly after flowering. This doesn't mean that the lines have to be short. That's just a characteristic of lines that flowered early and then stopped growth. We also have semi-determinate lines. Those are lines that in an indeterminate DT1 background have intermediate stem termination due to the DT2 gene. So the major genes affecting plant growth architecture are the indeterminate and determinate genes. There are independent alleles of the determinate version, DT1 lowercase, which is located on chromosome 19, linkage group L, actually near the E3 gene. So capital DT1 represents the functional indeterminate alleles specifying continued growth after flowering, while the 4 lowercase DT1s are all different missense alleles that condition determinate growth habit. The DT2 gene has recently been cloned and characterized at the molecular level, but the marker development is ongoing for that gene due to its very recent discovery. So some other things that we know about breeder selections in North America is that not only is there a distinction between the E1 gene, but also a distinction between the determinate gene. In north of the 36-degree line in maturity groups earlier than group 5 in North America, nearly all of our lines are indeterminate. South of that line, the ones that have functional E1 gene, nearly all of those lines carry one or another version of the determinate gene. This is thought to be due to the management of plant height and lodging in longer growing seasons in the south of the U.S. Okay, now I'm going to switch the presentation over to my student, Carrie Miranda. Okay.
Great. I'm hoping that everyone can see my screen. And I just thought I'd introduce myself for a moment for the people I haven't been able to meet face-to-face yet. So as you have probably saw on the link, you should have gotten a copy of a review paper that Kristen and I wrote. So at this point, I just want to talk about some of the literature background on the long juvenile trait and the many, many questions that are now posed now that we can see the data all together. And then after this, Kristen will talk about some more data, some actual data, and then I think this is a good flow for Alroy Kober's presentation. So, okay. Okay, so first I want to talk about what is the difference between the long juvenile trait and maturity. So in a 12-hour photoperiod, regardless of maturity classification, flowering is triggered before the vegetative stage is optimal, resulting in reduced plant height and node number. So when the long juvenile trait is present, flowering is delayed regardless of the shortened photoperiod. The long juvenile trait was discovered twice independently, once in PI 159925, and then another time in Pranagoyana and another variety SS1, which kind of go together. These were both observed on different continents. PI 159925 is a plant introduction line, whereas Pranagoyana and SS1 were just natural mutations in the Brazilian release variety Prana. And I was told that I don't pronounce that properly, so I apologize, Everson, for my strong American accent with your Brazilian lines. Both of these traits, when discovered, exhibited a 3-to-1 segregation, leading to the conclusion that the long juvenile trait was under the control of just a single recessive gene. In Ray's group, named the long juvenile trait observed in PI 159925 the J locus, whereas little j would be the long juvenile trait, condition delayed flowering, and Bonato and Velo in Brazil named this the long juvenile trait E6 when observed in Pranagoyana and SS1, whereas little E6 conditions delayed flowering. So already there are some questions that arise. First, are these actually two separate genes that we're observing, which would warrant the need for these two names? Or is it more likely that the long juvenile trait observed in PI 159925 and Pranagoyana are just different alleles at the same locus? And then, in which case, we wouldn't need the designation of two names. Alroy Kober did some experiments comparing the conventional lines to 3 different long juvenile varieties. So I'm hoping that my cursor can show you. At the bottom here, we have the conventional, as in the non-long juvenile Parana variety. And we see when the long juvenile trait is not present, that flowering will occur in around 25 days. However, when the long juvenile trait is present, and in this we have 3 different genetic backgrounds, they all exhibit a similar phenotype where they have delayed flowering until 45 days, which may be the more optimal vegetative stage. So I would also want to talk about the three different long juvenile lines shown here. Pranagoyana, which is the natural variant of Parana. So it's going to have the exact same genetic background as Parana, PI 159925. And this X line was created by using Pranagoyana as a long juvenile donor and back crossing it into an early maturing Harrisoi isolyme. So although they all have similar phenotypes at 12-hour photoperiods, which is the crucial photoperiod that we're discussing, I do want to bring some attention to the differences in phenotypes in earlier photoperiods, which you could say is a moot point because when are you ever going to have a 5-hour photoperiod? However, I think that it deserves some discussion. So if you compare the X line to Pranagoyana, which here will have different genetic backgrounds, but the same long juvenile allele, we can see that there is some slight variation. So I think that this shows that genetic background is going to play a role in days to flower. And then if you compare and contrast Piranha Guyana to 159925, which yes, they had different genetic backgrounds, but I think the question also arises, could this difference that we see, this phenotypic difference that we see, be attributed to different alleles at the long juvenile locus? In terms of genetic control of the long juvenile trait, in 2000 and 2002, this Brazilian group showed that the long juvenile trait is under the control of 2 genes. And they did this in Bragg, which I believe is maturity group 7, and they exhibited a 15:1 segregation of the long juvenile trait. And then in 2011, Alroy Kober observed that same segregation ratio again in the maturity group 0, the Harrisoin isoline, with Piranha Guyana as the long juvenile donor. However, when James and Lawn released their Melrose variety in 2010, they observed a 3-to-1 segregation, which using maturity group 3 Sprite and using 159925 as the long juvenile donor. So now I have even more questions. So why are we seeing this inconsistent segregation ratio? So the 15-to-1 segregation ratio suggests an epistatic effect between dominant genes, where you have to have, if there is one dominant allele at either of the loci, then the long juvenile trait will not be exhibited. So what I want to believe is that there's an epistatic interaction with one of the E genes, which when recessive will show the 3-to-1 segregation, which would just make the most sense. If the E genes are at least E1 through E4 are well characterized, they all play a role in the circadian rhythm, it would make sense that one of these proteins encoded by the E genes may be playing some kind of regulation with the long juvenile trait, which is most likely also involved in the circadian rhythm. However, the segregation ratios wouldn't make sense in, in the maturity 5+ groups, because all of maturity 5+ are going to have dominant alleles at all of the E loci. So you wouldn't be able to get the 15-to-1 ratio because you would never have the opportunity to introduce a recessive E gene. Now, this is complete speculation. This is me just doing Punnett squares, like textbook genetics. So I think Alroy Kober is going to comment on this, so if you disagree, go right ahead. There's also another possibility that a different gene or even a homologue of the long juvenile gene is present. And then this gene may be naturally recessive in many backgrounds, leading to the 3-to-1 segregation. It's the only other idea I have. Okay. And so I made this. This is all completely theoretical. I cannot stress that enough. This is not real data. But without known long juvenile markers, all selection for the long juvenile trait is based on phenotype. Unless there's someone here who has new information on that, my fingers are crossed that someone's going to say, oh yeah, we found markers for that like last week. I think it's important to understand that it may be possible to always select for functional E genes if always selecting for the most delayed flowering, which could be advantageous for areas close to the equator. However, in higher tropical latitudes, this may delay the vegetative stage too long, in terms of too long of a season or lodging, and which may not be desirable. Then different allelic combinations of the E genes could theoretically be used to create maturity groups suitable throughout sub-Saharan Africa, similar to what we have here in the States. And so I designated this as the different allelic combinations here, showing that the same way as you increase with more functional E alleles, that we have a delayed flowering effect here, which is how we have our later maturing groups. But instead of saying just the long juvenile trait, I designated that as just a recessive allele at the E locus, And in case there is a second gene that isn't one of the E genes that needs to be present, I designated that as recessive JJ. So again, that's, if you agree with me or not, that's fine. But I think it's important to note that the allelic combination of the E loci will play a role. And I also want to point out that there is some ambiguity when selecting for long juvenile and phenotype alone. And it could be possible to have a long juvenile trait in the early background as shown here, but overlap with some functional E alleles, as in here, this area here, which may skew segregation ratios, which again, this is complete speculation. I know that there are experts in the audience here who have been watching segregation ratios of the long juvenile trait for over a decade, so please let me know your opinion on that. Oh, and then here I only show functional E6 and non-functional E6, but since the molecular knowledge of this gene is not known, is it possible to have several different alleles of E6 which may have different phenotypes? So in short, has anyone ever observed different characteristics when using different long juvenile donors? And then last, just to convolute this all even more, what role do you think that determinate, semi-determinate, and indeterminate would play in all of this? So that's pretty much everything that I have. I know that Kristen has some data to share, so I'm going to switch it back over to her.
Okay, thank you, Carrie. I just have 2 slides I wanted to finish up this section with, okay, I seem to hit the wrong button for pausing that show my screen. Thank you for your patience during that. So this slide is a representation of some supplemental data that came out of the paper mentioned down here. Dr. Akrama, I was hoping would be on today, but I don't see him on the list. So this is data from his group where really they did an experiment to look at genotypic diversity on 36 soybean accessions from 3 countries. And I've taken from their supplemental data to make this figure so that the 36 lines are represented on this axis. They come from IITA in Nigeria, Embrapa in Brazil, and the SARI station in Ghana. And I have listed here in the first set of these 3 columns for each of the lines the days to flowering in blue, the days to maturity in red, and then plant height in centimeters here in green. And so what I wanted to show here is that there's quite a bit of similarity in the days to flowering and the days to maturity among these different accessions, which I will say were genetically quite distinct geographically in terms of the diversity analysis that they did. One thing that you can see that stands out, though, is that the lines from Brazil and from Ghana are much taller. And also I have a yield rank here, and this is not our traditional U.S. yield, but this is seed weight per plant. And so the yield ranking, might not work out in the end to the same as we would see it. But anyway, of the 5 top highest seed producers in this experiment, they were all from either Brazil or Ghana, and that then obviously correlates with the plant height being quite tall compared to the Nigerian lines. In addition, I want to point out that we have genotyping data on the lines that have the red underline here for the E genes and the DT1 alleles. And I would just like to say here, I guess, that we don't see a lot of difference in E genes in the lines that we've assayed. They all contain, for the most part, functional alleles of the E genes, so functional E1. In addition, all of the lines from Africa that we've genotyped so far are showing one of the determinant alleles, so they all have a missense, presumably determinant plant growth habit. So this slide is showing the data on an experiment that's in progress now in a low latitude environment in our winter nursery in Costa Rica. And here we've planted different lines that we know their E genotypes, their DT1 status. We think most of these are long juvenile, although that would be a preliminary analysis. And we're taking flowering time and maturity notes on those. At the very end of the discussion, I might bring up some of the flowering time data that we literally just got last night. But what I wanted to highlight here is the lines from Mozambique or Ghana that we have essentially have very, very similar E genotypes. This exception has recessive E2. They all have determinant alleles. We have an experiment here where we're growing indeterminate group 5s just to see what happens. And then a series of lines from Elroy Kober's work, determinate or indeterminate, that have the long juvenile gene brought in from this parent, Piranha goyiana. And then Dr. James may see his Melrose line here. We genotyped that, and although it was backcrossed, I believe, into the Sprite version that I don't recall, it has functional E1 alleles as well. So this is just a kind of a preview of the kind of things that we're thinking about, is putting our E genes, the determinate plant growth architecture, together with what we can figure out about any sources of long juvenile to understand flowering time, maturity, and eventually yield of those materials. So with that information, I think I'm going to end this part of the E genes, plant growth architecture, and long juvenile background, and switch to Dr. Kober for his presentation. And I'm just going to remind him to unmute himself. And I will mute myself. So Dr. Kober, please line up your presentation.
Okay. So I'm working with Agriculture and Agri-Food Canada in Ottawa. My work is with maturity group 0 to 000. I'm at about 45 degrees north latitude. To place that in the U.S., it's the North Dakota-South Dakota border. And I've got an emphasis on food type soybeans, especially those for the Asian export markets. I spent 3 years in Kabwe, Zambia, working with soybean research there, and so that's part of my interest in this group. Just to remind you what photoperiod sensitivity looks like. So this photograph illustrates the effect of 20-hour long days on soybean maturity. On the left-hand side, there's 150-watt incandescent lamps that are on early in the morning. They're off during the day, and they come on again before sunset and stay on and then allow a 4-hour dark period. So the plots under those lights are 3 meters wide. The alley is 3 meters wide, and you can see the effect of the lights reaching another 3 meters into that surrounding field. So this is just a visual example of long days delaying maturity. You saw an earlier picture of short days speeding flowering and maturity. We use this line to select for, or this nursery to select for lines that are photoperiod insensitive. Just a visualization of the association between maturity groups and photoperiod responses. So this is 2 pairs of isolines. The blue lines are Harasoy and an earlier version of Harasoy with the 2 null phytochrome A genes crossed into it. Then the other are the two Brazilian isolines for E6. So essentially we have a tropical long juvenile cultivar and 3 temperate lines, and the temperate lines are ranging in maturities from 6 to 00. temperate soybeans, and we've seen this before, essentially flower similarly at 11 to 12 hour photoperiods, and the photoperiods responses diminish as we get lines that are adapted to more and more northern conditions. So to associate photoperiods with some geography, I'm sending soybeans to Saskatoon, which is just over 52 degrees north. It has a maximum day length of 18.5 hours when you include civil twilight. Where I am at Ottawa, about 45 degrees, we have about a 17-hour day. In St. Louis, about a 16-hour day. In Kabwe, Zambia, where I was at 14 degrees south, it's about 13.75-hour day. In Accra, Zambia, or Accra, Ghana, at 5 degrees north, 5.5 degrees north, it's just over 13 hours. That's always including the civil twilight, which has been shown to include enough illumination or enough to trigger the photoperiod responses. So soybeans are not always planted to have their vegetative growth around the longest day of the year, but they can be planted when moisture is available or when they fit into the cropping system. And so planting out of the longest day of the year, then they will be experiencing shorter day lengths. Summer planted temperate soybean varieties are adapted to have some sensitivity to day length around the maximum day length at that adapted location. So you can see that the shoulder of the graph for each line, determines their sort of zone of adaptation based on the longest days of the year in that area. As it was mentioned, I introgressed long juvenile into a 00 Harasoy isoline. So I did it with two lines, Paranagoyana, the source of little e6. I actually developed back cross 5 lines, but in the first cross, the F2 segregated 15 early to 1 late. I also looked at the J source of long juvenile, and again in the first cross it segregated 15 early to 1 late. So in both cases I saw 2 recessive genes for late flowering when I looked at them under 12-hour photoperiods. So this is then following the maturity alleles, and this is genotyping that was done in Dr. Bill Yu's lab. So first the recurrent parent, it's little e1, the null version, little e2, little e3, it's also little e4, it's indeterminate. Then the long juvenile donor parent, and then we looked at the back cross 3 parent, the back cross 4 parent, and 4 long juvenile back cross 5 lines. You can see that the functional E1 was retained in all the selected late flowering lines, late flowering under 12 hours. The late flowering E3 and determinate growth habit are linked, and some of that was retained in some cases and not in all cases. And both those DT1 and E3 are loosely linked on chromosome 19. So a question I have from this introgression work is, why was E1 maintained through the back crossing, and why was the combination big E3 little DT1 mostly maintained, although it seems that we broke it in the back cross 5. And when I was looking back through my slides, I found this picture. So this is a back cross 2 F2 population segregating for the late flowering under 12 hours, and we can also see that it's segregating for the determinate growth habit, and you can see the difference, about 2 weeks in the flowering time difference between the long juvenile and the conventional ones. And it just struck me earlier this week when looking at this again that we've got this late determinate plant that appears to be as dwarf and short as the earlier maturing determinate plant. And when I see this dwarf determinate type in the field, that essentially means that it's a little e1, either one of the two versions of little e1 combined with dt1. So does this suggest that though the functional e1 was retained throughout the introgression, that perhaps the big E1 is not needed for late flowering? I think that's a question that needs to be answered yet. So I've been fascinated with the genetic control of soybean flowering and maturity time, having worked both in the tropics and here in Ottawa in the short season areas. And the question that occurred to me is whether we can have a green revolution type soybean. That is a soybean with a relatively fixed growth duration and with a wide zone of adaptation. So this is the photoperiod phenotypes as I see them. The black line on the bottom here is an early insensitive type, which is responding little to photoperiod. Then the red line is a typical temperate later maturing line, so as the days get longer it has quite a large response to photoperiod. So moving it north we get too late, too tall, too late. Moving it south we get too short, too early. Then perhaps this green line here is a long juvenile plus photoperiod sensitive line, and we really have to look at this combination and see if it has a relatively flat response to photoperiods. And then if we need to have delayed flowering to get a more full season green revolution type, can we find some true late maturity genes that are not photoperiod response genes, but ones that would delay maturity in a sort of a fixed way, so that we could have a range of lines from early to late that would flower and mature in 100 days or 120 days, basically regardless of where we planted them. This is some of the work that I have ongoing. So currently I have a cross between the E6 and the J lines and I have an F2 growing right now in the 12-hour photoperiod. I want to look at allelism between E6 and J and see if I get any conventional early flowering lines segregating out of there. I also have a plant a back cross 6 with an E3 long juvenile line to see if I can get it in an even earlier background. So I'm going to be looking for a long juvenile response in a little E3, little E4 one. And since looking at that photograph of what looks to me a little E1 long juvenile, I'm determinate. I think I'll have to make another cross and look for that combination. I'll also be looking to make a cross to end up with a big E1, little E3, little E4 determinant. This combination is one that will grow and mature in Ottawa, provide a relatively good yielding genotype, not quite as good as the conventional little E1 indeterminate types, but relatively good yielding, and to combine that with long juvenile. So with that, I'll turn it back to Kristen.
Okay, thank you, Elroy. All right, I'm just trying to stay organized here. Next on our list is Dr. Everson Calvo. So, Pete just reminded me because I actually forgot. I wanted to have a question and clarification session after each presentation, and I promptly forgot to do that after Carrie and my presentations. So I'll pause here and see if our panelists or attendees have questions and clarifications for either Dr. Kober or for Carrie and myself. So go ahead and unmute yourself if you have a question and ask it, and then for the attendees you can write in the question box. So questions about what we've heard so far or clarifications, please.
Kristen, this is Eberson. I have a question.
Okay.
On your presentation, the last two slides that you've shown,
Yes?
you showed some Embrapa varieties, and you mentioned that they basically did not differ on the number, the days for flowering, right?
Yes.
My question is, did you collect data on node numbers?
Okay. I have not collected data on node numbers yet, and the slide that I'm showing now was from a different work, and I think they did collect node number data. It would take me just a moment to look for that, so if you can just hang on. But so you must be saying that node number is important, and I think Brian and Randy may have some insight into that. Brian and Randy, do you want to say about if you're collecting, or George, if you're collecting node number data?
This is Randy. We haven't collected any node number data yet, but
yeah, this is Brian. We haven't either. I guess if Nicholas is on the line, he would be the one actually collecting it in Ghana, so maybe he could comment on it.
Okay.
Is Nicholas online?
Remember when you're going to speak to unmute yourself.
Yeah, Nicholas this is Pete Goldsmith. Nicholas is not on the webinar.
Okay, I'm looking back at the data from the paper with the 36 lines, and I do not see, I see branches, pods, seeds, mean 100 seed weight, biomass, lodging, nodule, height. No, I do not see node number. But
Okay.
Emerson, could you expand on what that might mean to us? Should we be focused on that?
I was just curious to see, because it's a little strange to me that they are all flowering basically at the same time. So I would expect to see some variation, because these lines, some of them, they do have, juvenile trait, different amount of juvenile traits, so I would expect them to vary. And so to me, it's saying that maybe there is another factor triggering the flower, another limiting factor triggering the flower a little bit earlier there.
Yeah, I want to say that this experiment that I have on the screen now was conducted, as far as I could tell, in Kumasi, Ghana, which I think must be 3 or 4 degrees latitude. Okay. So it was certainly very close to 12-hour day for this experiment.
Okay. Yeah, that makes sense.
So do you think that would make a difference to grow with that such low latitude near the equator versus maybe what you've experienced from 10 to 20 degrees latitude? Like, do you think that that small difference in latitude could be responsible for the difference?
I think it's a possibility, yeah. Because as we move, to closer to equator, we see a similar effect too.
Well, I think we could maybe get back to Dr. Kober's work with the different day lengths, and he's seeing a maximum difference in flowering time with that 12-hour day length, I believe. Is that right, Elroy?
Yeah, I saw a maximum at 12. 10 was close as well.
Okay. So quite short day lengths, but not unnatural. I guess you could say in a temperate part of the world, we can't grow things in such short day lengths because generally that's our winter. I mean, I guess there are some places that have short days and still non-freezing temperatures. In North America, where we grow soybeans, usually, you know, we just have one season. I know in other parts of the world where day length may vary, but not very much, that water is controlling the, water availability is controlling the season. Okay, so any other questions, clarifications? Courtney and Pete, do you have anything that came in? Because I'm not really able to monitor the question box myself.
This is Pete Goldsmith, Kristen. There's a question for Elroy from Tommy Carter. And Tommy asks, do you see a practical application for transferring the LJ type trait to the MG00 materials?
I'm not sure if there would be for the 00s, but I think it may be a way to have a double crop type variety in, say, a 1 zone where there might be enough time to grow some soybean after winter wheat.
So this is Brian. Kristen, the slide you have up, you said, perhaps you've mentioned this already, but were these the ones you genotyped for DT1? So the taller ones, did they have the determinate allele or were they indeterminate?
Everything that I've genotyped from Africa has one or the other determinate alleles.
Okay.
Which right now we talk about as being equivalent. I don't know if that's true or not. In my work with the maturity group 5s that have come mostly from Grover Shannon's program, we have seen one particular determinate allele, and this is the first time really where I've picked up the other determinate alleles. And actually the Dr. James line, Melrose, is the only time I've seen, I think it's the R62S allele in Melrose. And I don't believe Sprite, which is a semi-dwarf, so a determinate group 3, has that particular allele, so that's a little bit confusing to me. But everything from Africa that I have genotyped, and I say I, but I really mean Carrie, that Carrie has genotyped, and we've done it now by sequencing because we don't have assays for all of the alleles, but each one of the African lines has a determinate allele.
And the Embrapa lines are also determinate, right?
Okay, so all of those Embrapa lines that are quite tall, you know, compared to the Nigerian lines, I don't know what's going on with the IIT Nigerian lines.
Kristen, this is Randy. With your allele designations at the DT1 locus, do you know which one of those corresponds to the DT1T allele?
I had a comparison chart at one time, but I don't have that handy. So I don't know which one. Do you have any more background for me? Like, is that an allele that you see in the maturity group 5, 6, 7, and 8s?
Well, I don't know that it's the one that it exists in Peking and soy soda. I don't think that it's probably, as far as I suspect, it's not very widely distributed, but I don't think it's been checked very much.
Okay, so I have now up on the screen what I was talking about, and so this R166W is the one that I've seen in the, like, Dr. Chen's program in Arkansas and Dr. Shannon's program and also Dr. Pantalon's program. This is the allele that I see, although I haven't really gone looking for the other ones. And then this is the first time that I've seen this P113L come up. It just must be hanging around, although clearly the Ghana program has more than one allele that they're working with. And then, like I said, the Melrose was the first time I had seen this allele of DT1. Okay, I think, unless Pete, do you have any other questions from the box?
No, we're good
And
to go.
I'm just going to say,
We're good
because
to
I'm
go.
unmuted right now, that Pete, if nothing else, I will look through the slides that we have that we don't have a presenter for. If you can get George to do that, that would be great.
Right, we're
But
trying to
otherwise,
track George down now. We haven't heard back from him, but we're calling him.
okay, so I think then we can move forward. I have lost track of my organization here, but I think, correct me if I'm wrong, Everson Calvo, are you ready to go next?
Yeah, I'm ready.
Okay, I think we're going to go to you, Everson. And you're unmuted, so good.
I should share my, okay. You have me. Okay. I just, how do I share my window, my screen?
Okay, this is Kristen. So do you
Yeah.
see you have a...
You're good.
Yeah. Can you see my screen now?
Yes.
Okay. So, well, first of all, I need to say that, you know, my knowledge on Africa obviously is zero, and I also was trained with Dr. Romeo Kio, and just to mention that Romeo Kio doesn't really care about the E genes here in Brazil, so every time we talk to him, he says that E genes do not work here in Brazil, although I don't think that that's entirely true. I honestly believe that there are really interactions between the long juvenile trait and the E genes. And the third thing that I would like to mention is that after the excellent review that Christine and Carrie put together, I don't have much to add here, other than talk a little bit about the experience that the work that has been done in Brazil, okay, in terms of adapting soybeans to the low latitudes. So, close this window here. Okay. As you know, Brazil, we are one of the leading countries in soybean production, and when soybeans arrived here, they arrived in actually in the northeastern part of Brazil, but without success, and probably because of the absence of the long juvenile trait. So that was in early 900s, and it was about the beginning of the 60s that the soybean crops start to grow in southern Brazil, so under parallel 23. And essentially, varieties that were grown there were either complete lines that came from the U.S., mostly southern U.S., or populations that were developed in the U.S. and were used for selection here in Brazil. So when we start to move soybeans above the parallel 20, we start to run into troubles, and that was one of the situations where the farmers took the lead in doing that, and obviously without success. And then the research started to come, and nowadays mostly, roughly 60% of the soybean grown in Brazil is grown under latitudes smaller than 23 degrees Celsius. There are several reasons of this success. I just picked this slide here just to mention, again, the contribution that was made essentially by Dr. Hartwig, and here we can see Romeo Q as still a young guy working together with Dr. Hartwig, and they were on the first to study the long juvenile trait here in Brazil. Okay. This slide, on the picture on the left, we can see the difference that the trait makes in central Brazil. Okay. So the first thing that I would like to stress here is that first, all the experience that comes from Brazil is essentially to add the LG trait in determined backgrounds. And that was a choice made back in the late 60s and early 70s, essentially because was clear that it was difficult to control growth with indeterminate types. So all the work was done with determinate types in Brazil. The other thing that it's interesting to mention is that all the work was done actually here in Londrina under parallel 23. So we are located here just under the Capricorn tropic line. And the strategy used was to do the cross with potentially long juvenile trait source and plant these populations late September or the first 5 to 10 days of October, and essentially look for plants that would grow. And these plants were then selected based on the height, so everything between 80 centimeter to 1 meter would be sent to low latitudes to screen for local adaptation. So that was the strategy used to introduce these LG genes in soybean in Brazil. Obviously, the importance of the trait, higher yields. Obviously, the more nodes that you have, the higher the yields. Also, the wider planting dates. So we could start to grow soybeans not only in mid of November towards December, but start to anticipate the planting dates. And also, one thing that I believe it's very important, it was important here to Brazil and may be important to Africa, is that the long juvenile trait allows us to grow soybean for seed production planted in late December, beginning of January. And that's very important to central Brazil because we have too much rain during the normal harvest period of soybean, which is late January to beginning of March in central Brazil. So if you have long juvenile trait, you can get a better seed quality for the Cerrado area. And I believe that that's an important thing to Africa. So this slide, we show the relativity groups that we grow in soybean. So again, this is southern region. We are growing now essentially groups 5 to 6.5. We almost do not grow group 7, to 8 anymore here in southern Brazil. So that's another thing that happened. We start to shorten the maturity groups that are used for double cropping. And central Brazil, we essentially grow groups 6.5 up to 9. This is an interesting point, Christine. You mentioned that here in Brazil, we until 5 to 6 years ago, we essentially kept these 2 different regions, more, at least in our company, more separate. So we would breed things from southern Brazil, and we wouldn't cross much with materials from central Brazil. Nowadays, we cross among these 2 regions, so we do not have a limit anymore, because we start to rely more on indeterminate types, and that allows us to grow even groups 6.5 here in this region in central Brazil, especially on the higher altitudes of Goiás state and Mato Grosso state here. Okay, so that's something that has changed in Brazil in the past 5 to 7 years. Okay, so I'm just illustrating here in this slide. Until the 80s, we would grow soybeans from November 15 to December. This would be our planting dates. And nowadays, we do that in September up to November 15. The long juvenile trait helped, even in southern Brazil, to bring this planting season towards October. Especially, we could do that with long juvenile trait. We could anticipate the planting season until October 15. The indeterminate or semi-determinate types helped us to push this a little earlier, and now we can use the combination of semi-determinate or indeterminate trait to grow soybeans. We start planting on September 15th. Okay, so that's the trend here in Brazil. And because of this, as I mentioned, this push for double cropping, we start to rely more on the indeterminate and semi-determinate types. That started in southern Brazil. And this slide shows what happened from 2007 to 2011 here in Brazil. So we went from 80% of determinate types, and now we essentially are running close to 100% of indeterminate or semi-determinate types. Okay, so we are going through a process where we are learning to work a little bit with this long juvenile trait, different amount of long juvenile trait on indeterminate and semi-determinate backgrounds. Okay. So another thing that I would like to comment is that photoperiod insensitive genotypes didn't help us and to move soybeans to low latitudes. And essentially what happened was that these insensitive genotypes, the maturity group was too short, so we were not able to rely on these sort of genotypes to do this adaptation. As I mentioned, our experience is more with the determinate, but now we are using also indeterminate or semi-determinated types. And I'm illustrating here a case where we released 2 years ago a cultivar. It's a 8.0 maturity group. It's for the Cerrado area. And this material comes from a cross between Argentinian lines. One is ANTA 82, and this line here is in central Brazil is a 7.4 maturity group, but has a very limited planting date. Okay, you cannot plant this line in October, for instance. You have to start planting in November. So we developed this TMG variety that is the growers can plant anytime. So you can plant even in the beginning of October. And pay attention that this comes from a cross with other Argentina variety, 4.8 maturity group, and VMAX, which is a conventional variety here from Syngenta, also for southern Brazil, and it's a 6.5 maturity group. So I'm illustrating this, I'm bringing this example just to give the idea that we don't know almost nothing about this behavior of this indeterminate or semi-determinated types here in Brazil. So we are crossing semi-determinated types and indeterminated types, shorter maturity groups, and we are getting maturity groups a little higher, okay? So right now we are putting several long juvenile genes all in some specific indeterminated backgrounds. So we are developing isolines in indeterminate backgrounds to try to study this a little bit more. But unfortunately, we don't have data yet for this. Another observation that I would like to do is that the long juvenile trait, besides increasing the vegetative phase, it also, as you know, increases the reproductive phase of the plant. But one is able to select recombinants that has a little shorter reproductive phase if you want. So in a population, you can find some recombinants that allows you to introduce a long juvenile trait without increasing too much the cycle of the entire cycle or the maturity of the variety, okay? The other observation that we find here in our experience is that it's easier to move from higher to lower latitudes. In other words, when we cross, and I mentioned to you that we are crossing between lines adapted to central Brazil and southern Brazil. So when we do this cross, we can derive several lines from this cross to central Brazil, but it's much more difficult to derive lines from this cross to southern Brazil. So apparently, every time you come to south, there are other genes, and here I believe that the maturity E genes probably will be playing a role on that. It starts to increase the cycle of the cultivar, okay? And again, I already mentioned that all the work done was essentially done at the somewhat higher latitudes, but just using the earlier planting to select for the long juvenile traits. I mentioned that, again, that the review that you guys did was very good. I just would like to add 2 papers that came out recently that I didn't see there. A work done by Valeria Carpentieri, where she studied, and they have more data to publish. They did genetic studies on the long juvenile trait, using, for instance, the BR16, which used to be a very popular variety, proper variety here for southern Brazil, and 2 mutants selected in seed production fields that were late flowering or late maturity. And they were able to characterize and show that, for instance, the E92-7 has 1 recessive gene for long juvenile trait that adds 10 days for flowering, and the other line would have a second loci that would increase 25 days to flower on top of BR16. Also, they have shown that DOCO, which is a line that was adapted for central Brazil, conventional line, that may be a source for long juvenile genes for you guys in Africa, as it is a conventional material. These mutants that were selected in DOCO, they show that they have a pair of recessive alleles that control the long juvenile trait. Also in São Carlos, which is another variety, derived from Davis, has 1 recessive allele that delays flowering in 17 days. So I would suggest to look at this paper. I do have a copy if you want. The other thing that I would like to comment, another work, and this work was done at TMG. This work was done in 2000, early 2000, 2001, 2002, and was actually a dissertation published where one of our colleagues there tried to come up with an assay for determining the long juvenile trait in soybean. And essentially what he did was to grow soybeans during the winter in the greenhouse. And so he would supplement the light in different, and that was continuous light. That's an important point. I see that Dr. Kober worked with several different photoperiods there. But this work was done under continuous light for either 0, 14, 28, 42, or 56 days. And by doing that, one can separate and measure what we call the inductive phase, which is the phase where the plant starts to actually measure the day length, and by the difference of these days for flowering under a continuous light and days for flowering in zero supplementation of light, one would measure the juvenile period. Okay, so he did that, and he was able to measure for different genotypes here and to come up, and this, I apologize, it's in Portuguese, but I can translate here. These are days for flowering, the first column here, and the last column is the juvenile period. So one can find different genotypes with different amounts of juvenile period, and so essentially they may be either different alleles of the same locus, or different loci. The genetics that we try to keep to be practical. We try to make things easier. So we work with, like, say, 2 major genes. But again, we know that there are more, genes with smaller effect, and the recombination of these allows you to pull different degrees of juvenile period on this population. So essentially, that's what I would have to add to the previous presentation. And if you have any questions, I would be glad to try to answer it. Thank you.
Thank you, Everson. I know I have a few questions for you, but I wanted to start from our attendees for questions. So, Pete, do you want to kind of interpret the question and go ahead and give it to Everson?
Yeah, the first thing I wanted to note, we did hear from Nicholas, he's on the phone from Ghana as opposed to being on the webinar, and he wanted to answer Brian's earlier question about counting nodes, and he currently does not do that. He wanted everyone to know that. So that was the first point. And then Carrie had a question, and Carrie, I think you're on, so you could ask it directly. Okay, Carrie. Sorry, it looks like it looks like you do have a mic. I'll ask your question. Let me find it. What was that? Oh, yeah. Carrie's question was to Everson: did moving into indeterminate in Brazil improve yields?
That's a very good question. I actually do have a slide. I can pull it out, but okay, I did a sort of a compilation of data that was 5 years ago or so, where I gathered data from essentially strip tests conducted by different co-ops here in Paraná state, but several tests, and my conclusion was, yes, indeterminate types improved yield as long as you have good weather conditions. So in other words, and that was true for different planting seasons. Okay, 2 different years that I had data, and 3 different planting dates. But every time you have some stress, especially drought stress, and that, again, that's a compilation, I would say some empirical data. The tendency is that the indeterminate types would suffer more. I believe that that's not related to the growth type itself, because in fact, I believe that the indeterminate types, they should be more buffered towards this. That's my opinion. But I think that has to do with the source of materials that we rely. Most of the materials that come from Argentina, they are more adapted to maybe to soil, a different type of soil that we have here in Brazil. In Argentina, they tend to have better organic matter in the soil. So I believe that's an effect. That's an effect not of the determinate gene itself, I should say that the indeterminate gene itself, but rather from other genes that may make these materials more susceptible to drought stress. Okay, that's my personal feeling. I hope I answered the question. Okay.
Okay, thank you, Everson. I have several questions, and so maybe some of the other panelists are thinking of theirs, and I'll ask one, but maybe after one we'll see if anybody else wants to jump in. Kind of a general question that I have is, can you estimate how much of the Brazilian germplasm that's being utilized now has some form of what we are calling the long juvenile trait? So not just later maturity, but the actual long juvenile trait that shows up in those 12-hour photoperiods. What percent would you estimate of the materials grown in Brazil have that now? Because I'm a little confused between the very late maturities and the use of the long juvenile trait.
Okay. I would say, Kristen, that all the germplasm that we grow in central Brazil, that they are maturity groups from 7 above, determined types, they have some degree of juvenile trait. Okay? They all have 1, 2, or 3 alleles there that confer the long juvenile trait. Now,
can I interject here just a minute? Would you consider the variety Paraná to have a long juvenile characteristic?
Not really. Well, there is as I mentioned in my last slide, okay, you can see if you go to my, I need to project here, right? Okay, see, you do have, if you compare Paraná with, for instance, biloxi. Biloxi is something that it's a material that we say it would be a conventional type of line for us.
Okay.
So Paraná, it does have a degree of juvenile trait. I'll give an example. When we cross Paraná, which theoretically one would expect that it has almost no juvenile trait, and cross with another cultivar, Bossier, which also would be somewhat conventional trait, we can find in this cross, when we cross both, we can pull out lines. And I can actually tell you that probably 1 in 16 lines, you could pull out lines that are over 80 centimeters in growth and in lower latitudes. Okay, so that's why I'm saying, I mean, we try to give, every time we try to simplify things, but there are interactions in these genes, and some may be actually be the engines that are doing that. So you can, some material that you think there is no juvenile trait, when you cross them, you recombine them, and you find some juvenile trait there. Okay.
Great. Okay. If I can jump in, because that kind of segues to the question that I want to ask. So I think that's something I'm hoping to understand at the end of this workshop is speculation about interaction of the long juvenile trait in the E genes. And you showed a slide, I think from, I'm probably not pronouncing this right, Carpentieri Pipolo, where she showed different alleles for that's the same group that proposed 2 E genes, and then yeah, so you have here if you're recessive at one of the locus, and but then you have a functional allele at the other locus, that then you see different delays in days of flowering, which is actually a little confusing because they were the ones who first suggested the 15-to-1 segregation ratio, which would not prove this, but So I guess my question is twofold. Do you believe that there are different alleles for the long juvenile trait, or do you think that this might just be an interaction between long juvenile and the E genes?
That's a very hard question. I don't think we have, data. And again, Carrie, we do not dedicate much on study these things because at the end of the day, we are trying to look at, variety themselves. So
right.
I'm showing data that it's not our data, okay? It's Carpentieri and people, which probably here in Brazil, it's the person that studied this the more, in the past. And this work was done when she was actually started as a graduate student with Romeu Kiel back when Romeu Kiel was in Embrapa. This was actually done in early 2000, late 90s and early 2000. Okay, so I'm more a molecular biologist guy, and I really believe in interaction. I think we have different alleles. I bet we have different alleles on the J locus, and I bet we have interactions between J locus and the E genes. I really believe that, but it's more like gut feelings coming from a molecular biology. That every time we look at biochemical pathways and we see a lot of interactions, things are usually much more complicated than we think, right?
Right. Well, I completely agree with you. Actually, my background's in molecular biology too, so that's why this genetics thing is what I'm trying to wrap my head around, and I feel like the genetics of it isn't more consistent, but we don't have the molecular knowledge yet of the long juvenile trait to really completely understand the interactions, so I guess it's all just speculation.
But what is clear is that if you cross BR16 with the E92 line, you will pull out materials that has 10 days delay. And if you cross with this other line, it's a good source with a second loci probably with a larger delay. So you do have like 2 or 3 loci that are major loci there, and then you probably have another small loci that eventually when you recombine these small loci, you can actually get a pretty decent juvenile trait, and that's what we have done with the TMG 1180 line there. That's probably very low, small effect genes that we were able to put them together, okay, and get a decent juvenile or flowering delay, if you can say, in central Brazil. So I think you can go either way. Okay.
Tommy Carter has a question. Why don't you go ahead and unmute and go, Tommy?
Yeah, this is Pete Goldsmith. Maybe Tommy has maybe a problem with his mic. He did text in his question or his comment. He says, I would agree with Everson's comments on determinancy and stress. My work with determinancy suggests that determinancy versus indeterminacy trait is incidental to stress response rather than a main effect.
Okay, thanks for that comment. I have another question for Everson, and that is, you talked about the shift to earlier maturing varieties, and my understanding is that you are blessed with abundant rainfall conditions, so you can even triple crop in some situations, but I would like to know about how much can you expect to give up in yield for that short season variety or shorter season variety? Is there a threshold point at which your farmers are not wanting to grow those, or are they happy to take kind of whatever yield they get in that shorter season to push 3 seasons of crops a year, or what's the situation in the yield for the shorter season?
Okay. In southern Brazil, our problem is actually we have enough rain throughout the year. So our problem is the temperature. So we need to grow our corn. We need to plant our corn by, let's say, March 10th is our limit here. Okay. So we were able to move soybean to shorten the cycle, as I mentioned, and anticipate the planting date without sacrificing yield. Actually, we saw yield increase, okay, when we did that most of the time. Okay, the problem is in central Brazil. In central Brazil, we do have, let's say, an yield reduction, and that, I would say, farmers probably accept, I would say, anything between 5 and 7%. It's okay. A lot of times they double crop there, not only with corn, but also with cotton. So farmers that double crop with cotton, they tolerate less, they tolerate more yield reduction. When you double crop with corn, then because corn is not much of a cash crop in central Brazil, it depends on the year. So then they try to work more with a little larger maturity groups on soybeans. Okay, but I would say 5 up to 7% yield reduction. It's tolerated.
Okay, and then I have one final question for you, which is quite specific. You mentioned the use of semi-determinate varieties. I know that genetically that's been characterized, but just recently, molecularly, do you know your source for semi-determinate, and are you able to capture the indeterminate background that you need when so many of your Brazilian varieties were determinate? Like, how did you bring about semi-determinate, given that your germplasm collection, such to say, was so heavily skewed previously to the determinate background?
The reason is because we started to grow a lot of materials back in the early 2000s when the Roundup Ready beans were smuggled from Argentina to southern Brazil. They were essentially all indeterminate, and actually most of them, they were actually semi-determinate, but people didn't really know that. There was not too many people that could differentiate, the semi-determinate and the indeterminate. And so at that time, we start to use for crossing the Argentinian lines, and that's when farmers also start to push for even earlier planting dates. And as I mentioned, the indeterminate or semi-determinate help it to move from October 15 up to September 15. Okay, so, and nowadays what's happening, Christine, is that because we cross lines from southern Brazil with central Brazil, we are actually incidentally introducing the indeterminate types in central Brazil too, but that's not a necessity. It's more like a consequence because we end up crossing with southern lines here, southern varieties here. Okay, because you don't have much of a, okay, when we try to anticipate the planting dates in central Brazil, we didn't need much that. We just basically shortened the maturity groups in central Brazil, and that allowed them to start double cropping. Okay, so instead of planting maturity groups 8.5, we start to plant 7.5, and that allowed them to double crop with corn, for instance, in central Brazil. So there was no push for indeterminate. But again, it's happened more like incidentally, okay? And the other thing that I should mention is that keep in mind that central Brazil, we have high plains also. We have regions there that they grow soybeans in between 700 and 1900 altitude. And in this region, you also have a temperature effect, which delays flowering and maturity there. So it allows you to grow maturity groups of 6.5. You know, the same varieties that we are growing now here in Londrina, where I am, we can grow in parallel 15, for instance, at higher altitudes. And they are producing very well. Another thing.
I think we'll move on at this point, and remember we will have time for full discussion after our last presentation. So at this point, the thing that I don't know is whether Brian or Randy needs control of the screen. So if one of them could let Eberson know, I think he can change the presenter. If he's not comfortable, just tell me and I'll change. I just need to know, are we, next is either Brian or Randy or both?
So I'm going to go next.
Okay,
This is Brian.
Just put you on, Brian, then. Okay, so thank you, Eberson, and we'll switch gears now to the U.S. plant breeders' perspective on our SIL project in Africa. Go ahead, Brian.
So is my screen being shown right now?
Yes.
Okay. Yeah, so I'm working with Randy Nelson on this SIL project, and what I plan to do is just give a brief introduction of what we're doing, talk about some of our experience, especially with sending some germplasm from the USDA germplasm collection to collaborators in Africa, and then Randy will talk some about some of the experience in Ethiopia and how some of that material looked. So I'll more focus on Ghana, and what I'm going to be talking about with Ghana is some tests that Nicholas Denwar grew. So hopefully Nicholas is still online and he can answer questions or add anything if I misrepresent what he has been doing. So anyway, just to give you some background with the SIL project. As I mentioned, I'm working with Randy Nelson, and we're the U.S. Participants in terms of breeders on this project. We're working with 3 programs in Africa. So we're working with IITA. So IITA has their headquarters in Nigeria, but their soybean breeding program is really focused on Southern Africa. So the breeder down in Southern Africa is Hesham Agrama, and he's got a program both in Malawi and Zambia. And then we're working with a Bush Tefse, and he's at the Jimma Agricultural Research Center in Ethiopia. And then we're working with Nicholas Denwar at Sari, which is in northern Ghana. And so the 3 of them have breeding programs in Africa. And so our focus is really to work with these breeders in Africa who have the breeding programs there and to help them out to make their programs more effective. So the project is just getting started, and
I'm sorry, Brian.
We really need to have the
Brian,
display.
one sec. I'm interrupting, but can you put it on full slideshow?
Oh, okay.
Thank you. Sorry to
good.
interrupt.
Thanks. Yeah, thanks, Pete. Yeah, so one thing I have to say is that the project is just getting started, and both Randy and I have little experience in breeding or growing soybeans in tropical environments. So this is something really new to us. And one of the things that we're doing as part of the project is to try to bring in new germplasm into Africa. And so as I mentioned before, Randy has sent germplasm from the USDA collection to the 3 collaborators in Africa to see if they can identify germplasm that looks useful and adapted to African environments. What I'm just going to talk about is some work that Nicholas Denwar did in growing the USDA material in northern Ghana, and that's in Tamale, which is about 9 degrees north latitude. And this is from some data that Nicholas sent me in a report that I pulled out. And so what this shows on the x-axis is maturity group from germplasm that Randy sent him, and then days to flower on the y-axis. So what you can see is that at this low latitude, there's little relationship between maturity group and days to flower, which again suggests that the important thing at this short day length is the long juvenile trait. And you know, we don't know what genes these lines have, but perhaps some of these group 8s may have the long juvenile trait. I'm not sure. So that's just the relationship between maturity group and days to flower. And then we have days to maturity. Interesting, there's that group 4 line that has pretty late maturity. It would be interesting to follow up on that to see whether that potentially would have some unique genes to give it that longer days to maturity. Just to show some slides of what things look like. So this is some photos I took in Nicholas's field back in mid-March last year when I was there to visit the program. What you can see in the background, or way in the back, would be some lines that are from Africa, from Nicholas's program, that were planted on the same date as the material in the foreground. So the 2 rows you can see in the foreground are of a line that came out of Illinois, so it's a maturity group 4. So you can see very little growth, not a lot of potting, especially compared to some of the local germplasm. So that's a maturity group 4. This is a maturity group 8 line that came out of Georgia. Again, it's also very short. It was about the same size as the maturity group 4 material that we sent. This is a line coming out of Brazil. I don't know anything about it, its maturity, or where it's adapted to in Brazil, but you can see we've got more growth on that. I don't know if it's got the long juvenile trait or not. And finally, this is another line coming out of Brazil. What I did see, though, was that the Brazilian material did not have the growth, did not seem to have the potting on them compared to the local adapted material. So, you know, clearly there's more going on here than just getting the maturities right. So one of the things we're doing is we're making crosses between African germplasm and some U.S. germplasm, and our plan is just to develop populations that we can send back to Africa, and then have the breeders there start selecting to see if they can find lines that have the right maturities and are adapted to the African environments, but maybe have some better yield potential coming out of the U.S. germplasm. And for that, I'm grateful to Tommy Carter, who's done some crossing in North Carolina. We're also working with a breeder in Georgia who's done some crossing with their material. We've crossed with some of their material. We're growing F1 plants now, and we're hoping to get some F2 populations back to Africa for breeders to use. So that's all I have prepared. And then I guess if I can figure it out, I'll hand it over to Randy. So Kristen, is there a way, do you know how I do that?
There's a blue button on the right top part of your control panel that says Change Presenter that should give you a drop down. If you don't want to do it, I can do it.
Control. I'm not seeing it.
Okay, I'm going to do it then.
Yeah, you
Okay.
do it, Kristen.
Okay, Randy, you should be ready about now.
Yeah, there. Am I up now?
Looks good, Randy.
Can you see my screen?
Yeah, but
Yeah.
it's
Okay.
your,
All right, thanks.
Randy, it's not your slideshow screen, it's your, whatever they call it, your assistant screen, you know, so it's not showing the right.
Oh.
Switch your monitor, or, I mean, we can, it'll suffice. I don't want you to.
Well, see, I've got two monitors, and I guess
Yeah.
what you're seeing is my main monitor, and somehow my slideshow is... Oh, wait a minute.
If you go to the display setting, you can switch your...
There you go. Oop.
Yeah,
Is that
so go
good?
to the display setting at the top, and then you can choose to switch your views. Yeah, it's right there.
That work?
Yeah.
Yep.
Beautiful.
Okay. All right. Okay. Yeah, so I'm going to present the data that Bush collected, and I just got this data yesterday morning. So I was there early last November when this picture was taken, and that was, most of the lines were right then, but not quite. A bush is a breeder at Jimma Agriculture Research Center in Jimma, and that's at 7 degrees north latitude. So we sent the same set of material to a bush as we did to Nicholas. A bush had some problems with seed increases, and so he was testing then 60 of the 96 varieties that we sent, and he had 2 replications in a simple lattice. These are in U.S. maturity groups 3 through 8. We had some lines from Brazil, some from China, some from the U.S., actually a couple from Mexico. And then he had 3 of the common Ethiopian checks that that do well in that area. So this is some of the data that he sent. And what I presented here are all of the lines that had yields above the best Ethiopian check, and that turned out to be 10 lines. The things that I want to point out on here, I guess number one, this is 2 rep data from 1 year, so we need to be a little careful about how much importance we put on this data. The yields were high. They were much higher than I expected after seeing the plots. Whether this can be replicated in future years or whether these small plots are accurate in terms of the total yield, it seems to me the important message here is that some of this introduced germplasm was 20% higher in yield than their best Ethiopian check. So it looks like there's some potential for introducing some new and useful diversity there. If you look at the maturity groups, 4s, 5s, and 6s dominated in these high-yielding lines. There's only one group 8 here, which again was somewhat surprising to me. But when the International Soybean Program at the University of Illinois was conducting variety trials all over the world in lots of tropical and subtropical environments, back in the 70s and 80s, these kinds of results were quite common there, and I guess we still don't have a good explanation as to why they happen. If you look at heights, most of these are much shorter than we would like. There are only 2, this 5002T and Desha are close to 70 centimeters. Most of the others are certainly shorter than I would like to see varieties. The R8s, with 5002T, I've highlighted that. I think that's an error. I was hoping to get that corrected with the bush between the time I got the data and this morning, and I didn't get back from him. But I was there, and I'm sure that this was mature before then. But you can see for most of the rest of these introduced lines, there's not a great deal of difference in maturity. Their check was quite a bit later, and this FGOT is actually an indeterminate line. So 2 of their checks were determinate, 1 was indeterminate, and they're growing both determinants and indeterminates in Ethiopia. The other thing that I thought was interesting here is the 100 seed weight. These are all pretty high, so I don't know what that means. They're quite a bit higher than the Ethiopian check, and these are higher than what we would expect if we grew these in the U.S. Interestingly, this Sirek and Harbar were lines that we have in our collection. They were developed in Mexico. They were registered in crop science, and so they were put in our collection. They did relatively well when grown in Ethiopia. Some observations then. Missing from the top 10 list were all of the maturity group 3 entries that we sent, although 3 of them made the top 20. None of the Chinese varieties were in the top 10, although in all fairness, they were considerably older varieties than what we had from the U.S. 9 of the top 20 yielding lines were maturity groups 3 or 4. Again, not what we would expect. And then missing from the top 20 list was all but one of the maturity group 7 and 8 lines, and these are the ones that we had included. This is not all that we sent, and I think this Brazilian variety is probably pretty old. We don't have all of the best ones from the U.S. here as well, but these were the ones that a bush had seed to grow. This is what it looked like in the test. This is the Tennessee line, 5002T, maturity group 5. This is the Mexican line, maturity group 6. This one they had pulled up to thresh before I got there, and this is Ozark, but it looks like it's pretty well potted, and it yielded relatively well. This was we saw particularly in 3 varieties, plants were nearly barren, and most of the plants in the plot looked like this. There would be a few pods sometimes down close to the bottom of the ground. I guess talking to Elroy, this looks like something that they see when you get cold temperatures. What was interesting, though, is that it was maturity groups 3 and 4 lines that showed this symptom, and not the later maturity ones. And you would have thought that the 3s and the 4s, if anything, would have been tolerant to cold, would be more so than the 7s and the 8s. If you look at the average temperatures at Jimma, they're really very mild because their altitude there is, I think, about 1,800 meters. So they're pretty high. And in the box here I have, this is their, would be their main growing season. And you can see, at night their temperatures are getting down, this is the average, but 10 to 12 degrees, and in the daytime, not much over 20. So I think as we look to develop varieties in Africa, we're going to see some, even at similar latitudes, we're going to see some very differences in temperature. If we looked at the temperatures in Tamale, they would be much different than these, even though the latitudes are very similar. Finally, a bush took some disease data. I'm not sure what blight is. I suspect that it wouldn't be our bacterial blight. Most of our lines, I would think, would be pretty highly resistant to that. Although the scores were, were intermediate, they weren't exceptionally high. The rust scores were all moderate to very high. There wasn't really any difference among these. Bush's observations were that rust may not be causing damage that would be commensurate with these scores, because the rust tends to come in very late in the season, and so most of the the plots that still had leaves were really highly infected when we were there, but this was just in the last week or 10 days before they matured. And then on the last select here is anthracnose, and you can see again very little difference but very low scores. So this is a picture of a bush. He's a happy guy, and if there are any questions, I'll try to answer them.
We do have a question from the type-in that Pete can ask.
Sure. This is from a question from Cholani Wibade. Would Brian be willing to share some of the populations developed for Africa to be tested in South Asia?
I mean, it's possible if seed is available, as well as I would need to be sure all the people who contributed, the parents, are comfortable with it. If everybody's fine with that and we've got the seed, I mean it's certainly something we can consider and talk to you about.
I have a question probably for either one of you. How much do you think the temperature that you're seeing in Ethiopia is going to relate to other environments? So Ethiopia is pretty low latitude, but it has that unusual feature of being higher altitude, which affects the temperature. So how much of our thinking about breeding for adaptation will need to take into account temperature? Because as far as I know, in Ghana, they don't suffer from low temperatures pretty much ever. And I'm not sure about all of our places in Mozambique where we'd like to go, but do you see temperature as playing a big role in many programs, or just maybe at higher altitudes like the one in Ethiopia?
I'm not sure. I mean, I think my experience where we're working now, it seems like that Ethiopia is rather unique, and I think probably the biggest factor is just how low these night temperatures go. You know, I don't know that there are many places in the world where soybeans are grown where you would get, you know, consistently that low temperatures during the growing season.
So
and I think one of the things, even within Ethiopia, I mean, they have varieties selected for the high altitudes and then some for the moderate altitudes, and Jimma is one of the spaces or one of the places with the high altitude, and you can see big differences there. You know, in the tests that they would get from Paui, which is at lower, they would bring them up to Jimma, and they would be substantially later. So, you know, they would certainly select for earlier maturity there. How much they're selecting for, you know, for cold and how much difference that makes, I don't know.
So this is certainly something that is also important in Brazil. They have elevation differences. I wonder if Everson can weigh in on this and talk about how they deal with this in Brazil.
Yes, Ryan, here in Brazil, we essentially, we don't care about it. There is but I do have to mention that this picture of Maverick remind me of that same cultivar that I mentioned in my presentation. It's called Anta 82 from Argentina. That cultivar, when you plant it during the winter, let's say here under parallel 23 in the greenhouse, supplemented with light, you plant in cooler temperatures, we don't get any seed back. It's amazing. So my feeling is that we do have, like, you know, one or few genes, whatever, around, but most of the germ plants, here in Brazil, they do not have this problem. So once in a while we face this problem with, you know, a specific, and that's, so far it's the only cultivar that I know that has this problem. It's a cultivar that, as I mentioned, we brought from Argentina. And actually, I understand that this cultivar was a cross, a population was made in the U.S., was selected in Argentina and released by Helmont in Argentina. And we brought here to Brazil and has this problem. So we don't really care about it. I mean, we don't have this problem frequently, so there's no breeding effort for that. But apparently there are some genes hanging in there that, you know, may cause this effect.
Mm-hmm.
Okay. I have one quick question, if it's okay, Kristen.
Yes.
Okay. Randy or Brian, I'm curious to know if you had any problems with emergence, especially be interesting contrasting Ethiopia at the high elevation to Ghana.
The germplasm that was sent to Ghana had poor emergence, and we don't know if that's just because of of seed quality issues that occurred during transit, if the seed got heated up or something like that. But I mean, otherwise, what I saw for Nicholas's plots, he typically had decent emergence.
Okay,
Yeah,
Pete, you have a question?
I think that, I don't know for sure, but there were probably similar problems in Ethiopia. The plots that I showed, I had actually sent this seed to a bush a year before this project started, so we were looking at second generation seed there. But you know, as I noticed, he only had about 2/3 of those, so he had some trouble, you know, getting stands in those first generation lines.
Okay, I see a note from Pete that I am just going to put in here, even though it's out of place, that there will be a survey at the end of our meeting here. For all the attendees, please do that. Fill out the survey if you don't mind. And Pete told me there was a question.
Yeah, we had two questions. The first one from Tommy Carter at ARS. Did a bush provide any information on seed quality?
No, I don't have any of that yet, but it would be interesting to know that.
Second comes from Rita Mumm at University of Illinois. Brian makes the point that, and Rita, you can ask this if you want, and maybe you don't have oh, there you go. Okay. Rita, are you going to ask?
Can you hear me?
Yep, we hear you.
Okay. Okay, great. Thanks for the great presentations. Brian, as I looked at the pictures that you were showing and hearing you make the point about the adaptation of the U.S. material seems to be influenced by more than maturity and determinant morphology, etc. What do you think of possibly using an approach like Tanksley's advanced backcross to bring in a smaller dose of U.S. germplasm into the African background, say the varieties that were in the background that seem to be yielding and at least you know, showing high adaptation. Maybe like a BC2 population to bring in some of the germplasm in low dose from the U.S. background?
Yeah, I mean, certainly that's a possibility. I guess we're just trying to do something practical in terms of how can we get that germplasm, some of this new germplasm into the African programs as quickly as possible. I mean, what I would envision is we'll send them F2 populations or F3 populations. They can select out germplasm that's adapted and you know, flowers at the right time, has the right set of long juvenile traits and other genes, and then probably they'll cycle that through their breeding programs. So, you know, it seems like there's a good chance that they'll need to do a couple generations of selection, which would be kind of equivalent to what you're talking about. Developing the kind of structured populations that Tanksley has, I mean, I'm not sure how practical that is, because I'm not sure what germplasm we should focus all those resources on from the U.S. standpoint. You know, what variety should we use as the U.S. donor?
Sure. Yeah, I mean, and it could just be a sprinkling, but I think if you might have a better chance of favorable alleles from the U.S. germplasm actually showing through in the Ghanaian environment in backcross populations.
Yeah.
Just a thought.
I want to chime in here because I want to say this brings up really to me the fundamental point of this workshop was to explore what factors and, you know, as much as we could specifically what genes and alleles were necessary to create that optimum African adapted environment, or, you know, it could be country specific or region specific, we don't know, just so that we can move yield traits abiotic, abiotic stress traits, seed composition traits into the African adapted varieties and improve yield. So that's really the reason we want to have this workshop, is to find out what we can to develop this foundational system of what we know is necessary for optimum adaptation so that all those other traits can be moved in. So I think we are moving towards that. Are there any other questions for Brian or Randy, or if not, we can move on?
No questions from the audience.
Okay, so I think now we are at the point where we had hoped to have Jacob Tchagwa be our presenter for the African Breeders Viewpoint, but he was unable to attend. So I'm going to ask George if he's ready to talk about sort of African soybean experience, and if possible, also maybe present some slides that Jacob sent. So I'm going to make George the presenter. George Awuni, he's on the SIL project on the team with, on the, I don't know your MRA number, but I think of it as agronomy, and he and Dan Reynolds are the leads on that. Okay, I'm just checking here to see if that's working. So George, if you're on, could you unmute yourself, and do you have any slides of your own to show, or do you have a copy of Jacob's slides?
Yeah, I have some slides here.
Okay.
Are you hearing me?
Yes, we hear you, George.
Can you hear me? Are you hearing me?
Yes, we hear you, George.
Okay, is my slides up?
No, you need to show your screen. There's a button at the top of the control panel there.
Show control panel. I'm seeing show control panel.
See at the up at the top, there should be a blue circle with an arrow on it. In the yes, nicely done, nicely done, nicely done, excellent. You're on, and just put it on slideshow.
Oh, I'm clicking and yeah. Okay, is that right? Having trouble. Are you seeing the slides?
Yes, George. Yep, you're good. You're
Okay.
Good.
My name is George Awuni, working for SIL under Dr. Dan Reynolds, and I was in Ghana last year, 2004, basically conducting agronomic practices in the soybean in northern Ghana. Basically, this slide was showing is just showing the rainfall pattern in Ghana. As you move down south, you have a rainfall decreasing right from 700 to 1040, down to in Tamale area around 1100 to 1250. I went there with objectives of actually transferring the smart concept to farmers in sub-Saharan Africa, starting from Ghana, and then also trying to see whether we can increase soybean yields and definitely increase the incomes of farmers and improving nutrition and soil fertility. One thing about the soils in northern Ghana is that they are very low in soil nutrients. Specifically, we started with weather data collection. Over 30 years back data was collected, and then we also conducted soil tests in 3 locations, which we'll be seeing shortly. We conducted 3 experiments, seed germination tests. We looked at 15 varieties, and then we had a planting date test with 5 varieties, and then that was in Yankala, in Tamale, and then we had a variety trial which was in 3 locations, Wa, Boku, and then in Tamale. Boku is 11 degrees north latitude, and then we also had phosphorus inoculum test with 5 varieties. Now, so the mandate area of Sare is what we are seeing on this slide here. We have the Boku way in the north there, and then we have Wa, which is also in the circle in the, and then we also have Tamale right in the middle there. And this area has about 41% of the land mass of Ghana, and basically is a savanna zone. The Boku is a Sudan savanna, and then you have the Guinea savanna in Tamale, and then in Wa. Now for the soil test results, basically what we're seeing on this slide is the Yankpalada is Tamale. We see the structural classification down at the bottom. We took soil tests from the surface level and then subsoils down at the bottom, from 0 to 3 inches, which is the surface soil, and then from 3 to 6 inches, which is the subsoil. So we can see down at the bottom the classification. We have the low for Yankpala. We send these to ALL in Memphis for analysis. We also had a sample in Tamale for analysis, but the results I'm showing here is the analysis from Memphis ALL. So they give us these results: loamy soil for the Yankpala for the surface. Then you have the soft soil, which is a clay loam, and for the Boku, the surface soil is loamy sand, and then the subsoil was sandy loam. Now for the Wa, I had only a composite sample for testing, because at the time I got there, the land was already plowed, so I could not actually. It was meaningless taking, separate samples. So I just combined the 2 of them. That's why we have surface and subsoil together. All together, the result was that it was low. Now for the soil fertility rating and guidance, what we are seeing on this is the topsoil results. This is a busy slide, but I will not bother you with that. But just look at the colors. And then at the left-hand side, we have soil pH at the top, and then we have phosphorus, potassium down to nitrate, sodium. Now, and at the top, we see colors which is very low, low, medium, optimum, and very high. So for each bar along each test shows the rating. So we realize that we have yellow, red, which certainly shows that the nutrients in the soils are very low: phosphorus, potassium, calcium, magnesium, sulfur, and all that. With the only exception is zinc, which is optimum. Now for the subsoil, it shows the same pattern, that with the exception of magnesium and then the zinc, the rest of them are either very low, low, or medium. Now this slide here shows Boku, that's the topsoil, which shows the same pattern. This slide we also see is the subsoil for Boku, which shows the same pattern. Then for Wa, for the composite, it shows the same pattern. We only have magnesium and then probably zinc and iron probably looking better, but the rest of them all are very low or medium. Now this slide I just added this morning, this is women sorting out the seed. What I realized was that we did the seed germination tests, and the results on this slide that we see made me to let the women sort out the seed before planting. Now percentage germination was very low, with the exception of Nagba, which was above 80%, and then anidaso, which was above 80%, but the rest of them all was below 80%, which means that if the seeds are not sorted and planted, it's going to impact on germination and also impacting on yield. I would want to mention that 5 varieties were collected from Kumasi, that's CRI, which are also added into this to this 15 varieties that were tested. Now, for the variety tests, we had them located in the 3 locations. But what I'm showing here is from the Nampala station. I haven't had the data yet from the Boku and Wa, but this results is from Tamale. We tried 10 varieties, and each plot had a size of 22.5 meters squared with 6 rows in a randomized complete block. My treatments were basically the varieties, and then replicated 4 times. Now, sorry for this slide here because I was not able to make it very visible, but I just added it this morning. Now, if you look at the varieties, the range, I mean, there were differences in terms of 50 flowering days. We had the highest 72, and we had the lowest coming from Afayak and Sonda. I want to mention here that Songpong later was realized that it was a mixture of Songpong and Sonda. So I want us to treat that variety with caution. Maturity ranged from 122 down to 115. 119 was the highest, that's 72. I had no differences in terms of biomass, in terms of the varieties, and fresh biomass and then the dry biomass. For yield, for the variety tests, Kwashi came up looking very high, up to 3,000 kilograms per hectare, with the lowest coming from Songa. We slightly above 1,500. For seed weight, 100 seed weight, I had a Afayak above 12 grams per 100 seed weight, and the lowest coming from 722, slightly around 9 grams per 100 seed weight. I also looked at seed damage from the fields. I took 6 plants from each plot, from 3 plants from row 3 and then row 2 and row 5, and assess yield damage, taking out the seeds from the pots and then looking at the damage. And this is what I got from the field damage. Janguma came up in terms of the varietal trial with around 22% damage per plant. And then the lowest coming from Songa, somewhere slightly above 10, somewhere around 11% per plant. For the phosphorus inoculum trial, we use 5 varieties, and again, I want us to treat Songpong with caution. We have Janguma, Kwashi, Songa, and Afayak. I have plus phosphorus, plus inoculum, minus phosphorus, plus inoculum, minus inoculum, plus phosphorus, and then the control. The same design, randomized complete board design with factorial arrangement, with the same plot size and the number of rows, duplicated 4 times. 50% flowering is what we're seeing here. There wasn't any interaction in terms of the treatments and then the treatment of variety and then phosphorus and or inoculum. But the main effects had no significant difference in terms of the main effects with inoculum and or phosphorus. But the varieties themselves, taking the average across the treatments, inoculum or phosphorus, showed some significant difference, with Janguma coming up with 46 days to flowering. Not different with Songpong, which I said we will have to treat with caution. And then the lowest coming from Apayak. For yield, I had no interaction with the treatments and varieties. But the main effects showed some differences. Apayak wasn't significantly different in terms of yield with Janguma and Kwashi. And with Janguma, Kwashi, and Songda, and then Songpong, not significantly different. And we can see the statistics down at the bottom. Now when it comes to the treatments, phosphorus or inoculum, plus inoculum plus phosphorus showed higher yield, as you can see at the bottom there. There wasn't any significant difference between inoculum and phosphorus alone. And my control had 1610, which was the lowest. For 100 seed weight, there was an interaction. Slightly, the statistics for interaction variety and or inoculum of phosphorus was 0.05, and my P was 0.05. Even though this is slightly above, I consider that there's an interaction. So we see that phosphorus, the treatment has had an effect, an influence across board with the varieties. With Apayak, with phosphorus and inoculum coming up pretty high compared to Songda with the controls. Percentage seed damage had no interaction with the treatments, and there was no any significant difference in terms of the treatments, phosphorus plus or inoculum. But the varieties themselves showed significant difference, with the damage coming up, highly coming from Janguma and then Kwashi, with Apayak and Songda not showing any difference. For the planting date, the same varieties were used with the same design, but with 4 planting dates at weekly intervals. Same plot size with 4 replications. With this one, I'm only showing the yields here. Now, we see that the earlier planting gave us 1963, showing the highest across board, across the varieties, with the lowest coming from coming for the late planting dates on the 23rd of July. Now for the varieties, Apayak and Janguma and Kwashi did not show any significant difference. And again, Janguma and Songda did not show any significant difference. For 100 seed weight, I did not see any interaction between planting date and varieties. But Apayak and Janguma came up not showing any difference. And Janguma and then Kwashi did not show any difference in terms of weight per 100 seeds. Then the first planting date showed a higher weight in terms of 100 seed weight, not different from the second planting date. And the second planting date in terms of weight wasn't different from the third planting date. The last planting did seed weight was lower. There were a number of challenges in terms of controlling weight. So what I did was to, after 1 week of planting, I had to spray with a mixture of glyphosate and an alligator to control the weeds for the soybean to be able to spring up, for me to be able to control the later weed control. Now, weeding was also done probably around a V1, V2 stage, as you can see from this slide, sorry. The other challenge was insects. This insect here is the leaf-footed bug. It feeds on the tip of the plants just at the time of flowering, and that has an impact on the yield. So I had to do some spraying there. And we had also leaf rollers that were rolling the leaves, and then they would develop inside. These were not so much a pressure, but I needed to control them to be able to be at the safe side. Now I also recognized sting bugs in the fields. These were also sprayed with insecticides, Lambda Super 2.5. It's a karate that is a broad spectrum insecticide. There was also this leaf feeding beetle. This is one of the insects that I think I will have to watch seriously, or the station will have to watch seriously. The larvae, it has the larvae, and the larvae feeds. I guess I did not actually, but this is just a speculation. I think at the time of podding, it lays the eggs on the pods, and at the time of maturing, the eggs hatch and the larva penetrates into the seed. And what we see here is it feeds on the seed, and then they cause some talanin and defecate in the seed and in the pod. So this is a major concern that we'll have to look at maybe the next time I'm there and with scientists in Surrey. A major concern is also disease. Across the 3 locations, we observed disease in the locations. We sort of speculated. I'm not a pathologist per se, but I've taken courses in pathology. So we just sort of speculated with some technicians in the Surrey around anthracnose, and then the Cercospora, and then the Alternaria, and then some leaf blights. We can see my boss here in Boku. This is a Boku location where Dr. Reynolds is taking some pictures about some of the disease symptoms that he's seeing there. And now this is the impact of the disease. On the left corner we see some purple-like and then the black discoloration on the seeds. And then the disease also caused some sort of pores not filling up, as we can see on the right-hand side on the screen. There's a whole lot of them that I had to pick out from the pores. One of the things also, I'm not sure about this, but we also had an immature seeds. And I kind of speculate on sting bug effect, because sting bugs can also cause, I've read literature on the sting bug, and they can cause shrinkage of seeds and then the delay in maturity of soybean seeds. With that, I end here, and then if there are questions for me, I'll be able to answer them. Thank you very much for listening and your time. Right.
Okay. Thank you, George. I want to just say that questions for George we're going to handle in the questions box, and we can follow up with those offline. Either George can look at those, or we can do them after the webinar. So we will have this webinar recorded, and somehow they're going to make those questions available with the answers later. So I want to go ahead and move on actually to the rest of our program here and get a little bit back on topic to be looking at the major questions that we had that we were hoping to address, or that I was hoping to address in this webinar. So I want to kind of say that now we're going to transition to an open discussion. I'm not sure for the best way to do this other than to go ahead and speak with the discussion items that you have and mute when you're not talking. So the first question I want to try to discuss is, what are the most constraining features to soybean adaptation at low latitudes for optimizing yields? And from what I've heard today, I just want to summarize that things that I hadn't been thinking strongly about much earlier would have been things like plant height and node number as potential avenues for getting a better molecular understanding that we might be able to breed for in the low latitude environments. But I'd like to hear what the rest of the panelists and then any of the participants that want to weigh in here, what are we looking at as in our low latitude target environments? What is most constraining for optimum adaptation?
Okay, this is Eberson. Kristen, I would like to know, I just saw this presentation, and one thing that caught my attention was the amount of sand on this soil. That at least where George showed the results. Just to give an idea, here in Brazil, we don't recommend soybeans with less than 20% of clay. Okay, so and I saw that most of the areas that the experiment were conducted, they were like sandy soil, right? Is that the pattern in Ghana or in Africa? Because that's a major constraint.
Okay, sandy soil. Can anyone comment on that? Okay, so it sounds like maybe we don't know other than George's data from Tomali, or was someone else going to say something? No? Okay. So we can investigate what the effects are of sandy soil. Everson, why was that recommendation put into place?
Kristen,
what is the
sorry, there was a chime in at the last minute there. Carrie was saying that soil is also extremely sandy in Mozambique. And I don't know if Everson has any thoughts about colleagues who might be working in Mozambique.
No. The only information I have in Mozambique is that there is a Brazilian variety launched back in the 90s. It's called IAC-7. It's a 8.7 maturity group, and it yields very well in Mozambique, like 4,000 kilograms per hectare. But if most of the soils that you are targeting soybeans in Africa are sandy soils, and what is the regime, the rain regime in Africa? Let's say the presentation that we just saw in Ghana. What is the rain distribution there? Because here we really don't grow. We grow soybeans in 20% sand soil. 20% clay soil. Only in some regions in Mato Grosso where we almost have rain every day. Other than that, it's really hard to pull out decent yields of soybeans. And I was looking at Ghana, and the amount of rain per year is not that high. 12, 1400, but maybe the distribution, right? It's very concentrated, and maybe it rains every day.
So Emerson, so yeah, northern Ghana, the rain is really distributed only during the growing season. So they actually get basically most of the rain during 5 months when they're growing the soybeans. So it's a reason you don't recommend soybean production on these sandier soils is just because of moisture holding capacity is too low?
Yeah.
Or are there other reasons?
Well, there are two reasons. One is moisture, and the other is like the high temperature in the soils. It's very hard to establish a decent stand, right? Because it just kills the plant. You know, there's a lesion right by the, how do you say, the transition between the hypocotyl and epicotyl, that region. It just collapsed there because of the temperature. And so it's, you have to manage these sandy soils in Brazil. You have to rely on good cover crop with no-till to really be successful with soybeans there. So, because one thing that is striking me is that, you know, I'm trying to answer why doesn't this soybean varieties, for instance, Brian, that you planted there, it didn't grow as much. I was expecting them to grow more. So I wonder if there's some fertility limiting that's really holding these back.
Yeah. I mean, so these were on their experiment station, so they should have had decent fertility in the soils. And the Brazilian genotypes were much smaller than the local adapted genotypes. So whether there's just other genetics that we're not aware of that's needed to get the growth that's required for these environments, I don't know.
I think Nicholas Denwar is on the phone. Nicholas, do you have anything that you can add to this discussion about the soils? Okay, I'm not hearing him, so Pete, you might need to explore with that. Sorry if I cut somebody off there. Was there more on the soil fertility and the kind of unexpected or unexplained phenomenon that the Brazilian varieties were still not as, let's say, vigorous as the locally African adapted varieties?
And we haven't tested that many Brazilian varieties, so it may be just a matter of doing more testing and identify the best genotypes.
And so I'm wondering, in our crossing populations, are we better off crossing Brazilian by African adapted?
What's going to happen in this thing?
I think that's a good idea. And certainly one of the things was that by getting that Brazilian germplasm into Africa, we want the breeders there to make some of these crosses as well.
Sure. Okay, so other constraints that we might be able to get a molecular grasp on that I'm thinking about in terms of plant height, is there any consensus on should we at least evaluate some varieties with, say, a long juvenile characteristic and then indeterminate, which is what it sounds like is happening in Brazil, is that they're maybe moving towards keeping long juvenile in some aspect of what that means, but then letting the plants continue growth, or the semi-determinate, which would be a little bit trickier, but we actually could use a molecular approach for either one of those.
It will?
Yeah.
It's Andrew speaking from Australia. If I could just pull you out to a bigger picture first. We have quite a bit of experience in the low latitude tropics in Thailand, Vietnam, Indonesia, and Australia. So there's some commonality there with Africa, I presume.
Yeah,
The-
this was this seemed wrong.
Really the number one priority in any improvement program in those regions is to match phenology
What?
to the resources of the environment in
Question.
terms of the length of the wet season. So that's where the long juvenile comes in. And then the number two priority in most of those regions is actually seed quality. Getting decent quality planting seed and keeping it with high germination is a really big problem in the tropics, because soybean seed, and particularly high oil types from developed countries, deteriorate really fast. And so if you can't establish a good plant population, you're behind, you're likely to suffer from a very low yield ceiling right from the start in that cropping. So those two things are really important, matching phenology to the resources of the environment, hence the juvenile genes, and finding some way of ensuring good quality planting seed. Now the way we've done that in tropical Australia is to use the main wet season crop as the production crop, and then to grow a dry season crop over the winter as the main seed crop. So seed then is fresh and ready to plant the main production crop, for example.
Okay, thank you very much for that insight. I think in our target environments we have only the choice of one season because of rainfall constraints. So,
well, certainly in the tropics where it's hot, you're going to have to find a way of maintaining good quality or high germ seed somehow.
right, and so I know that in the southern United States where they run an early production season system, they are developing seeds under more tropical conditions during pod fill, and they have a seed quality issue, and they irrigate in cooler temperatures to do seed production. But I think our issues are there aren't necessarily cooler temperatures in some of our target environments. We know that seed quality will be an ongoing issue, and I'm not sure genetically at this point we're going to be able to address that. I do take into consideration the matching the phenology with the environment things, and I know, we're really pleased to see your development strategy published for Melrose.
We've moved on quite a bit from Melrose now. Now, we started doing this work about 20 years ago, and there is, or there should be, a resource that's available to the US of a whole heap of isolines that Henson developed, plus and minus the juvenile from that PI line 159925. We did some photothermal modeling of that of days to flowering, and it was pretty clear that there was that particular juvenile just conferred a delay in time to flowering, which was temperature dependent. It was about 400 and something day degrees, I think. And that was consistent across a very wide range of genetic backgrounds. So it's really just a case of inserting 400 odd day degree delay in 400 odd day degree photo-insensitive period at the start of soybean growth, and then tacking on the end of that almost the normal photosensitivity that's in the background. So the G by E you see at different day lengths is because plants are sampling a different sample. The isolines end up sampling a different sample of day lengths when you're growing them under natural conditions. Did I explain that very well? Possibly not. Because day lengths change through the growing season, the long juvenile ones are effectively sampling day lengths from a later window in the growing season than the conventional juvenile types. And if we mimicked that effect by just sticking a a longer true juvenile period at the start, basically the isolines came back together and behaved essentially identical, irrespective of what genetic background they had. Now that juvenile that we were using is about 8 or 10 generations removed from that PI line, so it's quite possibly only one gene got carried forward.
Okay, Pete has a question.
Yeah, there was a question from Carrie Clark about are there very many Brazilian lines that are not GMO that would affect our ability to use Brazilian lines as parents?
I think that question's for Everson, and he told me previously that his company, I think, is about 70% Roundup Ready and 30% conventional or something like that. So my understanding is there still are varieties. I'm not sure they're the latest, greatest varieties coming from his program that are non-GMO. I actually have a question for Andrew, which is, have you grown the Brazilian varieties in your locations? How do they perform?
Yeah, we've grown quite a lot. We had quite an active program of importing Brazilian material before the GM stuff became more dominant, because we're GM free here. And so we've evaluated must be a couple of hundred in tropical Australia and also in Vietnam and Thailand. And southern US material too. We do find that differential in height there as well. So if you have a maturity group 8 of the traditional type, traditional photosensitive type, quite often they turn out to be very short things. And we've always kind of attributed that to the southern US gene pool becoming very short in stature when we take them into tropical environments, even if phenology is not quite as severely constrained as, even if phenology is not too bad, you still get that severe reduction in height. That's definitely not as severe in group 5s and 4s and 3s. So for example, the old US variety Williams was actually quite successful as a parent in a lot of tropical Asia. But anyway, yeah, so maturity group 4s and 5s tend not to become quite so short in height when we move them into the tropics, whereas a lot of those 8s, for example, become very short indeed, even if phenology is not too bad. And then if you compare them to the locally adapted material in Vietnam, for example, there's quite a big difference in height. So they must have selected lines that produce much higher vertical growth, even in very early maturing material that is still determinate.
Okay. We have limited time for our workshop due to the webinar, and I believe it's going to end, the recording at least probably, I don't know if the whole thing will end in probably about 20 minutes, so I think if it's okay with everyone, what I'd like to do is just sort of speed to the end at this point. I know that we all have our contact.
We've done crosses with materials such as Paranaguayana, Tropical from Brazil, sources of the long juvenile out of the Philippines and Indonesia. A lot of their land races, the very old varieties, have long juveniles, and the source that we have. My gut feeling is that the Paranaguayana one is different from the one we were using and does tend to confer a longer juvenile period. And the other thing was that we were trying to develop a series of photoinsensitive type probe genotypes that were in basically a FISCB5 background, which is photoinsensitive, with various different juvenile genes. but I really stopped work on that about 15 years ago because I've had to work on other things. So we did think that was possible at one stage, and certainly Elroy is kind of continuing that work, I believe.
Okay, does anyone want to weigh in on evidence for an allelic series for the long juvenile gene? Is it more likely to be kind of a plus minus? You have it or you don't. It has its effect in possibly different genetic backgrounds. Is there any evidence for actual independent loci? Seems like from what I heard presented today, we aren't quite at the answer to that. What we do know is that long juvenile is successful in creating adaptation in tropical environments. Seems like it's fixed pretty much in the Brazilian materials now. It's possibly fixed in the African adapted materials. Obviously Australians.
I don't think that TGX material has juvenile genes in it.
When we grow the TGX material in a 12-hour day length,
I know it's
very
late, but
late flowering.
yeah, but I don't think it's the conventional juvenile that we've been using, because you can actually get that to be mature quite quickly if you're growing under very short days.
How would you define very short days?
Oh, less than 10.
Okay.
Yeah.
Okay, and we don't know really the pedigree of those TGX lines. I think that Brian and Randy are exploring, trying to understand.
I think some of them came from the old, because they were promiscuous nodulators, that was derived from Mamloxi and Avoyelles originally via some intermediate varieties.
So what do you think the source of that is? Some Chinese landrace?
Either Mamloxi or Avoyelles.
Okay.
Andrew, what was that second variety name?
Avoyelles, A-V-O-Y-E-L-L-E-S.
Oh, okay, yeah.
You have that? Because we've certainly still got it here.
Yeah, I'm sure it's in that collection.
Yeah. I think Mamloxi's a black one.
Okay, and then how close are we to a linked or perfect molecular marker for long juvenile? I think this would be very useful if we're trying to
The
make
marker
cross.
about 20 centimorgans, I believe.
What chromosome?
I don't know. This is getting to be close to 20 years ago for me.
Okay. There is a paper that I found that uses, I think, rapid technology and has a linked marker. I think it might have been from the Argentinians. I can't remember exactly, but anyway.
A colleague of mine put it over Henson's isolines, and it worked pretty much, I believe.
This is the Henson marker?
Yeah, the Henson long juvenile, yeah.
Okay, so from this,
there's not in the literature somewhere because we would have picked it up.
the one paper that I could find, like I said, had a rapid marker with no linkage group type of test or anything, but the little bit of work that I did seemed to point to chromosome 4, possibly. I think a better characterized marker could be nice in this case, because at least for me in Columbia, Missouri, I really can't grow these African lines from seed to seed. I can't get them to flower and mature in my growing season. So it limits some of the work we can do here, although we have the use of winter nursery, which has turned out to be really good to deal with these things. Okay, so then just some general thoughts on what efforts, what research directions can we apply to develop high yield potential, and this isn't short season versus full season varieties at low latitude. And I know, Andrew, you said you had some work in Vietnam and Thailand. I've heard that Vietnamese farmers won't take a soybean variety over 90 days. So are you working with short season and full season varieties there? And can you give us any insights into how we might move forward in a tropical environment?
The model of soybean cropping that was coming out of ABRDC was very short season cropping. A lot of regions of Southeast Asia where rice is the dominant crop, they have a cropping system where they'll relay or triple crop, so 3 crops in 1 year. So they're very reluctant to go to a soybean that is more than 90 days, as you say. So it depends where you are and how long their wet season is, whether they'll grow a longer duration type. So that's a constraint in tropical Southeast Asia, is that they tend to be tied into the rice cropping season, system. And your only opportunity for increased productivity there is to push high plant population, high harvest index, high inputs, and maximize yield that way. But I think the African picture is a bit different because you're certainly not growing in rotation with something like rice generally, are you?
No. But maybe there would be a companion season or a low-risk season. I think each environment will be different, but we do have some focus on a smallholder. So we want to find opportunities just to get some experience maybe with soybean production that could turn into medium-scale farming, but we do want to do some we want to see the smallholder be successful with soybean as well as a commercial scale.
Yeah. So are there good sources of inoculant?
We are working on that as part of the project as well.
Yeah.
I do have a question then. In your work in Southeast Asia with the 90-day system, are you back then to a U.S. maturity group 4 or 5?
Oh, that was the other thing I was going to talk about. Once you get into the tropics, the maturity group system really doesn't work. The GBE for time to flowering and maturity is quite large, and that's in some parts related to GBE for planting date in times to flower. So you don't get the nice or relatively nice linear maturity group thing that happens in North America particularly. And that's because you've got a reasonably tight planting window, seasons that change reasonably quickly in terms of temperature and day length. And so varieties do fall out into a nice ranking that matches a maturity group. Now, in Australia, we don't have that sharp change in seasonal conditions for temperature. And many of our growing regions run north-south rather than east-west, so we need varieties that are adapted over a wider range. And so the maturity group system doesn't really work for us either. So I would just feel a little bit cautious about trying to aggressively or very firmly push the idea of a maturity grouping system in the tropics, because I think you might struggle to make it work because of the very large GBE.
So just local selection for yield then is going to be as successful as targeting any kind of specific maturity?
If you want to move advances made in more temperate regions into the tropics, you can do the long juvenile conversion of maturity group 4s or 5s, for example, and they'll convert into something like 110, 120 day bean into the low latitude tropics. So that's certainly a quick pathway to move those advances in. However, you do need to keep an eye on the disease resistance profile of those varieties you're converting, because some of those won't have all the resistances you need in the tropics.
Dr. James, what do you think about taking an earlier maturing, even just the example of like maturity group 2, North American variety, and then introducing the long juvenile trait? Do you think that that would be a way to achieve a short season?
Yeah, well, that's what we've got a variety called Moonbee, which is basically a maturity 2 genetic background with the juvenile, and I'm not sure whether it's determinate or semi-determinate, but it's the earliest maturing juvenile that we currently use. In your situation, it would probably be about 4, maturity group 4 with the juvenile present. So the background of that must have been closer to 2, I think. So yeah, that works nicely because you've got that photoinsensitivity, and you can go to very early planting dates. So our growers are starting to do what they do in Brazil and move to very early spring planting dates, and having the soybean mature kind of late summer rather than at the start of winter.
So is that still a 110 to 120 day life cycle?
Yeah. It does depend a little bit on temperature, because they're still temperature dependent even if they're not very photosensitive.
Okay.
If you move very early, you're starting to get into much cooler temperatures, which can delay flowering.
Okay, now...
While we mentioned temperature, I would agree that the temperatures, minimum temperatures below 15 can often lead to that sort of sterility.
You've seen that too?
Yeah. So in Vietnam, in the north, they'll grow a winter season crop, and temperatures there are often low enough that in a lot of material you can get that kind of sterility. It's pollen sterility. So some of the more tropical varieties, such as the landrace variety landrace varieties out of the Philippines and Indonesia, you get pollen sterility at about 15. Something like Fiscabee 5, it's down towards 10, for example. Fiscabee is a progenitor of some of the Canadian material. They came out of northern Japanese material originally.
I'm sorry, I missed what you said just a moment ago about Fiscabee 5.
Fiscabee has a much lower threshold for cold temperature sterility than many of the tropical varieties.
Okay.
That's published somewhere about 30 years ago, I think.
Okay. Okay, I think I'd like to wrap up now with an opportunity for any unanswered pressing questions about meeting the objectives of this workshop, or things that people would like to add or say. We have a few minutes just to sort of open up to, no framework here, just if anybody wants to make a short statement or have a last remaining burning question. I think we can wrap up in the next 5 or 10 minutes when our time will end. So I'm just going to open it up here for questions. Remember, you have to unmute yourself.
I'll just make a comment. I thought Andrew's comment was very, very good, and I think this strategy of trying to pull out this, to put the long juvenile trait on the, let's say, American varieties, just paying attention to the disease package that the Africa may need. I think it's an interesting approach, definitely. And it's more or less what we have done in Brazil, you know, in the past here.
Okay, that's a good comment, and I think that Carrie and I are thinking about ways of manipulating the long juvenile trait, especially in combination with different E alleles, which is really what the American lines would contribute in terms of maturity genes. Hopefully they would contribute yield as well.
Yeah, I don't know if that was clear in my last slide that I showed, that I kind of think that if we just use different E allelic series and then add in the long juvenile trait, if it's true that 159925 has a different allele phenotype than Paranagoyana, then that would be interesting. But the way I kind of see it is just that we could make a maturity group system work in Africa by combining the allelic series that we know of the E genes with the long juvenile trait. And so I'm curious to hear if anyone agrees or just like that's totally not going to work.
Have you guys seen that the paper that I mentioned? I actually took a picture of it. It's I think the first and only work of maturity groups in Brazil, published in Crop Science in 2011, I think, by Ali Prandini.
Who's the first author on that paper?
It's Ali Prandini.
Okay, no, I was thinking of Destro. There's a good review by Destro.
Oh yeah, there's this one called The Stability of Soybean Genotypes and Their Classification into Relative Maturity Groups in Brazil.
Yeah.
Is that the one you're speaking of?
You've seen that?
I'm literally, I've got it in front of me right now on my computer. But the first author on that is Jose Cavassim?
Yeah, Cavassim.
Yeah.
I'm not sure. I think the first author, the one that I mentioned, is Ali Prandini.
Okay, then this is a different paper then.
It's a Crop Science, 2011. It's Ali Prandini is ALLEPRANDI. No, and Ali Prandini. D-I-N-I, Ali Prandini. I think it's interesting to look. I kind of agree that it's hard to adapt these maturity groups, but it's working reasonably here in Brazil. So there is the effect of altitudes, but I think you adjust as time goes by. You adjust it, and I think it's interesting to read what they found out.
Okay, any additional thoughts? I know we have some questions coming up.
Can I just comment on one other constraint in the low latitude, lowland tropics, and that's high temperature. If you start getting mean temperatures up around 30 degrees Celsius, so it's max plus min over 2, soybean really struggles. You start losing the ability to accumulate biomass at those high temperatures. Now that may not be a problem in places where it rains a lot. I'm not familiar with the environment in much of Africa, but it would certainly be a problem in places like the Middle East where they irrigate their soybean but temperatures are very high indeed.
Do we know anything about maximum temperature? George, what's the maximum oh, George had to go, I think. Maybe not. George, if you're on, can you address the maximum temperature during the growing season in Ghana?
I'm not sure yet, but I think that the temperatures in Ghana range between 80 to 90 degrees Fahrenheit. That is just around 28, 30.
Okay, so it might not be in that upper range.
So it's not in the upper range, yeah.
Yeah, like in Arabia, I think they have much higher
Yeah.
daytime temperatures.
Yeah, they sound like good temperatures for soybean.
Okay, we do have a question that I can see, and I'm not sure who it comes from. It says, what does Andrew think about promiscuous nodulation? Did they look at that in Southeast Asia?
Yeah, we've looked at it in Australia with that TGX material out of Nigeria, and we looked at it in Vietnam. It's not promiscuous outside Africa, I don't think. So it still requires nodulation in Australia, and it still required inoculation in Vietnam, so it didn't confer an advantage.
Right. I don't think I'm aware of publications demonstrating a defined advantage for those promiscuous nodulators in Africa. I know they did a lot of breeding work, but I'm not sure the yields really increased with those
I think
lines.
they're probably promiscuous with African strains of Rhizobia, but they're not promiscuous with ours, certainly.
Okay. I wasn't sure that even though they were nodulating with those native strains, that it was effective nodulation.
Oh, I believe so.
Okay.
Yeah.
And then I see...
Some places. I was aware some places down in down towards Zimbabwe, for example, not recently, but they were no longer promiscuous down there, so it possibly they weren't promiscuous everywhere in Africa either.
And actually, I have a question for you. Do your farmers have to inoculate every season they grow soybeans?
No, it's good for 20 years or more once you've grown a crop.
Okay. And then we have
the advice is to inoculate because they quite often will go 5 or 6 years without having a soybean crop in the same field.
Okay, I have another question that I'm not sure who it originates with. It is, do Australian lines have rust resistance?
Yeah, we have two. We've got two major genes we're using for rust resistance, and we think our variety Bunya has a sort of a rate reducing effect, so it delays the onset of rust by sort of 3 to 5 days, which in itself is not particularly significant, but we hope by combining the other the immunity genes, or whatever we want to call them, we'll have a bit more tolerance. But rust is not a major problem for us because generally our temperatures are too high. Rust is more a cool temperature, moist conditions pathogen, not a hot, dry pathogen. And most of our environments are hot and dry, not cool and moist.
Okay, I think judging from the time that our time is probably up for this webinar. I want to thank a number of people for their participation in making this webinar, which, as I said, was my first, very successful. So I want to remind you we were just trying to develop a core of knowledge for maximizing soybean adaptation, really as a platform for those low latitude target environments in Africa. Thank you for contributing to that. Pete, Courtney, and Carrie helped get this going and really did a lot of the legwork. Our panelists, Elroy, Brian, Randy, Everson, we had hoped to get Jacob Chicagua, but thank you, George, for stepping in there. And then we had some VIPs that I was hoping to kind of stimulate discussion with, and they're listed here, although I saw that not all of them could attend. So I just want to close by really acknowledging the Soybean Innovation Lab project and the funding from USAID for making this possible. I think we can continue to work together to develop resources and research for development in our target environments. So I'm just going to close.